Hello everyone, I'm Priyanka and today we will talk about the equipment used in the clinical microbiology laboratory. First we have analytical balance. An analytical balance is a type of a balance that is commonly used for measurement of mass in sub-milligram range. Talking about working principle, these type of balance are made with a measuring pan enclosed in a transparent covering that prevents small particle or air current from getting collected on the pan. An electric analytical balance uses the force necessary to counteract the mass rather than measuring the mass itself. An electric an electromagnet is used to create a force required to achieve a balance with the mass of the substance and resulting force is displayed. Talking about use, as they are highly precise and based on the advanced technology, analytical balance are used in laboratory for the effective completion of tasks like weighting test material and sampling amount, formulation, density determination, purity analysis, quality control testing. As you can see in the figure, one side we have analytical balance, like traditional analytical balance. Now presently we use electronic analytical balance. Next we have autoclave. Now an autoclave is a pressurized chamber used for the process of sterilization and disinfection by combining three factors: pressure, time and steam. Talking about working principle, autoclave use steam as their sterilization agent. The basic principle of an autoclave is that all the items within the autoclave come in direct contact with the steam for a particular period irrespective of the nature of material, whether it's a liquid, plasticware or glassware. The amount of the time and the temperature depend on the type of the material being sterilized, and the increase in the time of the cycle allows the shorter periods. Talking about use, autoclave are mostly used for sterilization of medical and laboratory equipment with the capacity of sterilizing a large number of materials at once. They are commonly used for preparation of culture media during laboratory applications. So here you can see a vertical autoclave that are mostly used in laboratories, and on the other hand we have horizontal autoclave. Next we have Bunsen burner. Bunsen burner is a standard tool used in laboratories named after Robert Bunsen. It is a gas-fueled single open flame. Talking about working principle, this burner is made with a metal tube on a flat base with a gas inlet at the bottom of the tube, which may have a adjustable wall. On the other side of the tube are openings which can be adjusted with the color to control the amount of air that can enter. Once the burner is connected to the gas source, the gas is forced by the gas pressure so that the gas reaches the top where the flame is ignited with a match or a lighter. Talking about use, it is commonly used for processes like sterilization, combustion, and heating. In medical or microbiology laboratory, it is commonly used for micro-loop sterilization. So this is a burner. Next we have centrifuge. A centrifuge is a device that allows the rotation of an object about a single axis where an outward force is applied perpendicular to the axis. The laboratory centrifuge is a motor-based and allows the rotation of a liquid sample, resulting in the separation of the component of the mixture. Talking about the working principle, A centrifuge works on the principle of sedimentation, where the high speed of the rotation causes the denser particle to move away from the center while the smaller, less denser particles are forced towards the center. Thus the denser particles settle at the bottom, where the lighter particles are collected at the top. In a laboratory tabletop centrifuge, the sample tubes are aligned at an angle so that the particles have to travel a shorter distance before they hit the bottom. Talking about the use, the primary application of the centrifuge is the separation of particles suspended in a suspension. It can be used for the separation of cell organelles, nucleic acid, blood components, and separation of isotopes. So this is the centrifuge. Next we have colony counter. A colony counter is used to estimate the density of a liquid culture by counting the number of CFU, that is colony forming unit, on an agar or culture plate. Talking about principle, this instrument can accommodate different sizes of plates which are scanned on the top with UV, white light or fluorescent illumination. One can accomplish the counting either manually with the power with a touch pressure or with a digital counter. Talking about use, a colony counter is primarily used for counting the numbers of colonies present on the culture plate to estimate the concentration of the microorganism in liquid culture. So this is the colony counter. Next we have deep freezers. Talking about the working principle, deep freezers are based on the principle that under extremely low temperature, there is a minimum microbial growth for the protection protection and the preservation of the different substance. Based on this principle, we can even preserve culture over a long period of time without any change in the concentration of microorganisms. Talking about the use, a deep freezer can be used for the preservation of the different things used in laboratory for a long period of time. Deep freezers are used in laboratory to store, preserve medical equipment, food items, blood samples, medicine and injections, etc. for a more extended period of time. So this is a deep freezer. Next we have hot plate. A hot plate is a stand-alone appliance used in the microbiology laboratory as a tabletop heating system. Talking about the working principle, unlike the traditional way of producing heat through the fire, a hot plate produces heat by the flow of electricity. On a hot plate, electricity runs through the coil which have a high level of electric resistance. The resistance in the coil converts the electric energy into heat energy which causes the coil to release heat. Talking about the use, in laboratory, hot plates are used to heat glassware and their components. They are used over water bath as in water bath might be hazardous in case of any spill or overheat. So this is a hot. So next we have hot air oven. A hot air oven is an electrical device that is used for sterilization of medical equipment or samples using dry heat. Talking about the working principle, hot air oven is a type of dry heat sterilization which is performed on dry material and on substances that do not melt or catch fire under high temperature. There are two types of hot air oven based on the working principle. Forced air hot air oven is a type of hot air oven. The heated air inside the oven is distributed throughout the oven with a fan. This prevents the rising of the hot air towards the top while keeping the cool air at the bottom. This allows the adequate heating of the material inside the oven. Next we have static air hot air oven. This type of the oven, the heat is produced by coils present at the bottom of the oven with no fan. The hot air rises and does not allow the effective sterilization of the material. The equipment inside the oven acquire heat and pass the heat towards the center on the layer at a time which allow for a effective dry heat sterilization. Talking about the use, hot air oven can be used to sterilize materials like glassware, metal equipment, powders, etc. It allows for the destruction of microorganisms as well as the bacterial spores. So this is the hot air oven. Next we have incubators. An incubator is a device that is used in the laboratory for the growth and maintenance of microorganisms and cultures. Incubators provide an optimum temperature, pressure, moisture, along other things required for the growth of microorganisms. Talking about working principles, the incubator is based on the principle of maintaining a proper atmosphere for the growth of microorganisms. Incubators have a heating system that allows for the temperature within the incubator to be adjusted according to the type of the organism cultivated inside. Similarly, they are provided with adjustment for maintaining concentration of CO2 to balance the pH and humidity required for growth of organisms. Variation of incubator like shaking incubator is also available, which allow for the continuous movement of the culture required for the cell aeration and solubility studies. Talking about the uses, incubators have a wide range of applications including cell culture, pharmaceutical studies, metallurgical studies, and biochemical studies. Incubator can also be used in the stem cell research area. So this is the incubator. Next we have laminar air flow or laminar hood. Laminar hood is a closed device primarily for processes or instruments sensitive to microbial contamination. Talking about the working principle, a laminar hood is made up of stainless steel, avoiding joints and corners to prevent the accumulation of bacterial spores. This device creates a sterilized environment with the flow of sterilized air through a high efficiency particulate air filters and short-wave ultraviolet germicidal lamp that sterilize the workstation. Laminar air flow has to turn on 15 minutes before to ensure complete sterilization, and the workstation should be cleaned with the ethanol before and after use. Talking about the uses, laminar hood is commonly used to conduct processes that are sensitive to contamination. It is used for experiments related to plant tissue culture and for the experiments of genetic transformation. So this is a laminar air flow.